it's here.
Unless it's here? Ah.
That's the clicker, but that should also have...
Yeah, it does.
Here it is. Okay.
Yep. If I could ask the panelists to please take their seats, we are going to start. Because otherwise, why would they need translation?
And then if they raise your hand,
Yes.
and then if they do...
Our translator's over there.
Okay.
If not, she will go home eventually.
Okay. Hello. Quiero preguntarles si algunos de los que hablan español necesitarían traducción para la sesión de la tarde. Tenemos disponible la traductora. Así que, por favor, si necesitan traducción, acérquense a ella. Gracias.
Good afternoon. Welcome back. Just a word of thanks again from the FDA for our patient panelists from this morning. Your opinions were very rich and wonderful to hear. And I think as reviewers, and I think also folks working in the pharmaceutical industry would join with me in saying sometimes we don't get to see the faces connected to the treatments that we might be working on and developing. So it makes a huge difference for us. And so we really appreciate you being here. And you really have made a difference. Also, it's now time for you to get to ask us questions. So I want to begin by asking our scientific panel to introduce themselves. And I think we'll start with Barbara on that end.
Hi, I'm Barbara Herwell, Centers for Disease Control and Prevention, Parasitic Diseases Branch, and I'm honored to be here. Thank you.
I'm Karen Byrne. I'm a medical epidemiologist. I'm at UCSF now. I used to be at CDC, and I do research on Chagas disease.
Hi, I'm Danong Chen. I'm with Metronomics.
Tom Smith, Medical Team Leader with the Division of Anti-Infective Products, CDER, FDA.
Maria Allende, Medical Officer with the Division of Anti-Infective Products, FDA.
Sumati Nambia, Director, Division Anti-Infective Products, CDER, FDA.
And hi, Ed Cox, Director of the Office of Antimicrobial Products, CDER, FDA.
I'm John Farley, Deputy Director of the Office of Antimicrobial Products.
I'm Isabella Ribeiro. I'm an infectious diseases physician, and I had the Chagas disease area at the NGI, Drugs for Neglected Diseases Initiative.
I'm Sheba Maymandi. I'm the director of the Center of Excellence for Chagas disease that's based in Los Angeles, affiliated with all of you, but a Department of Health Services facility.
I'm Joe Turner, the Deputy Director for Safety in the Division of Anti-Infective Drug Products at CDER, FDA.
Good afternoon. My name is Sergio Sosa Estani. I'm the Director of the National Institute of Parasitology, Dr. Mario Fatala Chaden, Minister of Health of Argentina. Thank you for the invitation.
I am Alejandro Sigman, Head of the Laboratory of Molecular Biology for Chagas disease that depends on the National Council of Science and Technology in Argentina. Thank you.
I'm Louis Kirchhoff. I'm an infectious disease physician at the University of Iowa, and I have a longstanding interest in Chagas disease.
I'm Kiliana Suzash-Wojcik. I'm epidemiologist at Bayer Pharmaceuticals in Germany.
And Jaime, would you like to introduce yourself?
Sorry. My name is Jaime Alche. I am head of the parasitology services in Buenos Aires Children's Hospital.
Great, thanks very much. So we'll have a couple of formal presentations and then a panel discussion, and that'll be the format for the afternoon. Our first speaker is Karen Byrne, who is a professor of epidemiology and biostatistics at UCSF. She is a former CDC investigator who's worked in endemic areas of Latin America in disease transmission, diagnosis, treatment, and biomarkers of disease progression. And she's also published comprehensive reviews of the disease in both NEJM and JAMA. So Karen, we're delighted to have you here today. Thanks. And this is a pointer if you need it.
Good afternoon. Let me figure out the technology here.
Sorry. Yeah, so this should advance your slides. Let's see. Yeah. Okay. There you go. So it's this one, to go back, to go forward. Right. Laser.
Great. So you see the same picture again. Those of us who work in Chagas disease have a lot of fun putting together our slides because there are so many amazing historical pictures from the days of Carlos Chagas. By now, I think you all are aware of the major Trypanosoma cruzi transmission routes. This is to remind us all that the transmission is through the feces of the vector, which actually means that it's not very efficient. Most people who are living in an endemic area are exposed to the vector and to the parasite many times over the course of their lives. And then, of course, the other modes of transmission which become much more important outside of endemic areas and as vector control becomes better: congenital, transfusion, transmission, and through contamination of food or drink. I want to remind all of us that Chagas disease in many ways is a public health success story. 25 years ago in 1990, before the first international control effort in the southern cone, which means the southern part of South America, the estimated prevalence of infected individuals throughout the Americas was 18 million, and there were an estimated 500,000 new cases per year. The most recent estimates, which were just published by PAHO, now estimate that there are just under 6 million infected people and about 40,000 new cases per year. And I'd also like to point out that the way some of these estimates are arrived at is that there's sentinel population surveillance in most endemic countries of Latin America. And usually that's done by doing serosurveys in children under the age of 5. For example, in Brazil, there are nationwide surveys of children under the age of 5, because those are new infections. This comes back to some of the discussions this morning about the early chronic phase and what we in the Chagas world mean when we say that. And what we mean is that essentially children with infection have of necessity been infected sometime during their lifetime, so more recently than, for example, an adult in an endemic area who could have been infected any time, in the last decades. And this becomes important when we start talking about response to treatment and the assays that we can use for that. This is extremely rough. This is from the most recent PAHO estimates. But you can see that there is T. cruzi throughout the continental Americas, and the most affected countries are Bolivia and Argentina, with Ecuador, Paraguay, El Salvador, and Guatemala as sort of the next tier in terms of prevalence. I also want to say that I come from an endemic country. The U.S. is not a non-endemic country. We don't, as far as we know, have very many
human new infections in the United States each year, but it's certainly, there's certainly an enzootic cycle that's established across the southern United States. And we have at least 11 competent vectors in the United States and many infected reservoir hosts. And there, we can go into details on this, but just so that you're aware, all across the southern United States, if you test raccoons or possums or wood rats in the southwestern United States, you'll find anywhere from a few percent to 40% infected. I'm having trouble making this work. Better. And this speaks to some of the questions that came up this morning in terms of the blood supply in the United States. So in the U.S., most of the U.S. blood supply has been screened since the beginning of 2007. And the current reports on the AABB website, which is open access, are that there are just a little bit over 2,000 confirmed infections picked up through the blood supply. And you can see that it's almost every state in the nation, but with concentrations in areas where we would expect to find large numbers of Latin American immigrants. So, as we've mentioned before, the locally acquired Chagas disease burden in the United States is undefined because we've never done large-scale serosurveys, and those would be very expensive. There have been 7 locally acquired vector-borne human infections documented clearly since 1955, 4 of those in Texas, and 1 each in California, Tennessee, and Louisiana. But other than the Louisiana case, all of these were acute infections that were picked up usually because an infected triatamine vector was found near the person who turned out to be infected. Clearly, there are many more infections that go undetected unless someone gives blood, like the donor that we heard from this morning. An extrapolation from a study of 16 blood donors who were apparently infected in the United States suggests a prevalence of 1 in 350,000 donors. That's compared to somewhere between 1 in 5,000 and 1 in 10,000 donors from Latin America. We know that there are more than 20 million people born in Chagas disease endemic countries of Latin America who live in the United States. And some years ago, we estimated that that would extrapolate to about 300,000 infected immigrants based on the T. cruzi infection prevalence in their countries of origin. And what Sheba was talking about this morning I thought was very interesting, that she's finding about 1.5% prevalence when she goes out and does community-based serosurveys, and that would come out to exactly the same estimate. And so that actually gives me quite a bit of confidence in this estimate that we did in the past. But this is infected immigrants. And in case series, including that Sheba's group did and another that was done in New York City, somewhere around 13 to 16% of non-ischemic cardiomyopathy in Latin American immigrants can probably be attributed to T. cruzi. So we know that there are many people in the United States who have Chagas cardiomyopathy. Most of those are probably never diagnosed as being from Chagas disease. I'd like to just very briefly go through the natural history of the disease. You've heard this once this morning from Maria, but I think it doesn't hurt to go through it again. So after vector exposure, the incubation period is 1 to 2 weeks, and then a person, or an animal for that matter, that has been infected has what's called the acute phase of Chagas disease. I'd like to point out for those of you who are clinicians that the incubation period can be very much longer in someone who acquires infection through transfusion or organ transplant. And so the index of suspicion has to be high. Fewer than 1% of acute infections are thought to be detected. This doesn't mean that they don't occur. It's just that they're not diagnosed as being from Chagas disease. And this is because most people don't have something like the Romagna sign that this little girl has that would tip you off that what this is Chagas disease. Most of the symptoms, if they occur, are mild and are very nonspecific. Fever, malaise, hepatosplenomegaly, atypical lymphocytosis, sounds just like mononucleosis. Very rarely, someone in the acute phase will have severe symptoms. Acute meningoencephalitis and or myocarditis. So these are rare, but when they occur, they're associated with a very high mortality rate. They tend to occur more either in the extremes of age, so in very young individuals especially, or in people who are immunocompromised. So you're more likely to see this, for example, in an organ recipient. And the hallmark of the acute phase is what we call patent parasitemia, which means parasitemia that you can see on microscopy. And this can either be on a wet prep where you actually see the parasites moving, or it can be on a stained smear like the one that you see here. It also means that in the acute phase, molecular assays, PCR-based assays, have extremely high sensitivity. And so this is a very good tool in the acute phase. Definitely having technical problems. Congenital T. cruzi infection. So this is an acute infection, but an acute infection in a neonate. If you look at meta-analyses of studies of cohorts of infected women, you find that a median of about 6% of infants who are born to infected women will be infected themselves. But like Chagas disease in general, most of these are mild or asymptomatic. Most of the babies we see in Bolivia are actually asymptomatic. Rarely you can have a severe acute phase in the baby, and the mortality from this is high, but we don't see this very much. And so what that means is that babies need to be screened. So first you need to know the mother's infected, and then once you know the mother's infected, the baby needs to be screened. And it's actually not trivial to screen the baby, because the test that's usually used in Latin America, what we call the micro method or microhematocrit method, is microscopy of a concentrated cord or neonatal blood specimen, and the sensitivity of that in a single specimen is only 50% or less. So most babies are going to be missed on their first specimen, which means you need to do multiple specimens, and that's not very acceptable to parents. PCR has higher sensitivity, but it's still not 100%, especially in cord blood. And that's probably because the parasitemia usually rises after birth, and so you may have better sensitivity at 1 month or 2 months of age than at birth. So you can see already this is a really complex screening program. And so, for example, Argentina has probably the best screening program for congenital Chagas disease, but even so, the last evaluation I saw made it clear that many babies were being missed because many babies were not brought for their final follow-up at 9 months when serology can be done, and that has 99% sensitivity because you have to wait till 9 months when the maternal IgG has disappeared. And so this is a difficult screening program. Without treatment, within about 8 weeks, people then pass into what's called the chronic phase of T. cruzi infection. The parasitemia falls steeply because of the immune response. So people have to mount essentially an inflammatory immune response to control the parasite. And the acute symptoms, if there were any, will go away spontaneously. At this point, the blood smear will become negative, and PCR sensitivity is variable. And I know that Alejandro is going to talk a lot about sensitivity of PCR and how to maximize it, so I won't go into any details on that. The diagnosis at this point relies on IgG serology. And there are a number of different tests. There are several different kits for enzyme-linked immunoassays, ELISAs, the immunofluorescent antibody test, something called the TESA blot. In the United States, the reference laboratory is at CDC, and CDC will do reference testing on people who need it for Chagas disease diagnosis. And because no one of these tests is perfect, we generally confirm with at least 2 different tests that the person is truly infected, just like you do in HIV infection, for example. Although you can't see the parasite in the blood, these people are infectious to the vector, can transmit congenitally or via transplant or transfusion, and can reactivate if immunosuppressed. So you've already heard this morning the indeterminate form of the chronic phase is someone who has no cardiac signs or symptoms, no GI signs or symptoms, and a normal electrocardiogram. They may have some subtle abnormalities if you do more advanced testing. At this point, we really don't know the prognostic value of those more subtle signs. And some experts, especially in southern South America, will require negative barium enema and barium swallow as well. We'll talk about that in a minute. And as you already heard this morning, this is a lifelong infection in the absence of treatment. Most people will remain asymptomatic in what's called the indeterminate form for the rest of their lives. They'll never have symptoms. And about 20 to 30%, the estimates vary depending on the studies that you look at, will progress to either cardiomyopathy or gastrointestinal Chagas disease or both. So what is Chagas cardiomyopathy? You've already heard a lot about this morning. You've already heard from people what their experience is when they have this disease. This is a really bad heart disease, and it has many different manifestations. Usually the earliest signs are conduction system defects, so what we call a right bundle branch block or a left anterior hemiblock, so the conduction system is not conducting normally. And later on, higher degree atrioventricular blocks, so people can develop complete heart block, and that's usually associated with a severe slow heart rate, severe bradycardia, possibly with syncope, or even with sudden death. This, the cardiologist can speak to this more than I can, but one of the things that's seen in Chagas heart disease are both slow and fast heart rates, so brady and tachyarrhythmias. People get sinus node dysfunction, what we used to call sick sinus syndrome, and severe bradycardia, but they also can get ventricular arrhythmias, they can get atrial flutter and fibrillation, and so many different symptoms along with the arrhythmias. Apical aneurysms are actually quite common in people who have advanced Chagas heart disease, usually in the left ventricle, and they can develop clot, thrombus, in the left ventricle or in the aneurysm, and because of that, they can develop strokes. And then the end stage, as you've heard from several of the patients this morning, is a dilated cardiomyopathy and congestive heart failure that eventually may not respond to medical treatment. I want to touch very briefly, because this is talked about in some of the studies of treatment, on classification schemes for the severity of Chagas cardiomyopathy. And there are really just two components to this. The first are the characteristic EKG changes, the characteristic electrocardiogram changes. So you've already heard those: right bundle branch block, left anterior vesicular block, bradycardias, other heart blocks, atrial fibrillation or flutter, left bundle branch block, and then signs of cardiac insufficiency and eventually congestive heart failure. And so those you can do that either with a chest x-ray that shows heart size or with an echocardiogram where you can measure the left ventricular end diastolic volume and the ejection fraction. Don't be intimidated by this. This is just 4 of, I think we're probably up to 6 or 7 different schemes, but they all really follow that same kind of pattern. So usually class 1 or class 0 or class A is indeterminate. So people have a normal electrocardiogram and no signs of congestive heart failure. And then the second class, or, so 1, B1, B, are only EKG changes. And then 3 and 4, or C and D, are people who have cardiac insufficiency and then frank congestive heart failure, or cutoffs that correspond to that based on ejection fraction. And this really has to do with the prognosis of Chagas cardiomyopathy. So signs of poor prognosis include, as you might imagine, complex ventricular arrhythmias, global or segmental wall motion abnormalities on echocardiogram, sustained or non-sustained ventricular tachycardia, and then the signs of congestive heart failure, an increased left ventricular end diastolic volume and decreased ejection fraction. Sudden death from Chagas disease can occur either earlier or later in the course of disease. Early sudden death is usually from a ventricular arrhythmia or complete heart block, less commonly from an embolus. And then mortality, late mortality, tends to be from intractable congestive heart failure. I won't spend very much time on gastrointestinal Chagas disease, but this is the other major manifestation. It's less common than Chagas heart disease and tends to be seen in southern South America and to be very, very rare in northern South America and Central America. Not non-existent, but rarely seen. And even in the southern cone, where it's most common, it seems to be less than 10%, in contrast with heart disease, which is more like 20 to 30%. And both for the esophagus and the colon, which are the two end organs most commonly involved, it involves disordered peristalsis and eventually a dilation of the organ, so you end up with a megaesophagus or a megacolon, or sometimes both. We think that the geographic patterns have to do with strain differences in the parasites. That's been hard to prove directly, but certainly there's a difference in the prevalence that you see in the northern part of the hemisphere versus the southern. And the treatment is largely surgical. As far as we know, treatment with antiparasitic drugs will not affect the outcome in terms of gastrointestinal disease, although admittedly we really have no data on that. In the United States, I think one of the first things that brought Chagas disease to our attention was an episode where three recipients of organs from an infected donor developed Trypanosoma cruzi, and at least one of them seems to have died from it. This was in about 2001. We now know, with 15 years of data, that T. cruzi transmission risk varies depending on which organ is transplanted. There are pretty good data for kidney transplants, which is the most common transplant, that a minority of people who receive an organ from an infected donor will actually develop T. cruzi infection. 13% in a US cohort, 19% in an Argentine cohort. For liver, it seems to be around 20%, although admittedly the data are very sparse. There have been some hearts transplanted from infected donors, and 3 of the 4 who received hearts from an infected donor developed T. cruzi infection. Recommendations that were published in 2011 based on these data recommend that kidneys and livers from infected donors can be used, preferably with the knowledge that they're coming from an infected donor, and that presumptive treatment is not recommended, that what is recommended is serial monitoring with PCR, and this is pretty much always done with CDC, which can provide that PCR monitoring, and then a lot of advice over the course of the monitoring of the patient. And what we know since 2006, when there have been quite a number of recipients monitored this way, is that there are very good outcomes with early detection and treatment. The other thing I'd like to touch on very briefly is reactivation in immunocompromised hosts, because we've also seen this in the United States. There are 2 major settings for reactivation, either a T. cruzi infected patient who receives a solid organ or bone marrow transplant, so clearly a person who has a heart transplant for end-stage Chagas cardiomyopathy, and then in the setting of HIV T. cruzi co-infection. In the transplant recipients, the most common manifestation of reactivation is an acute myocarditis, and this is usually picked up when endomyocardial biopsies are done in the course of the monitoring of a heart transplant recipient. And again, this has a good prognosis with prospective monitoring and treatment. So it's a matter of really the physicians having a high index of suspicion. In HIV co-infected patients, the most common manifestation is CNS disease, so a mass lesion in the brain and or a meningoencephalitis, and this is associated with a very, very high mortality rate. Acute myocarditis is the second most common manifestation of reactivation, and treatment in these settings involves antitrypanosomal treatment and optimization of antiretroviral treatment. It's really unclear at this time what the role for antitrypanosomal prophylaxis is, because there haven't been that many of these patients, but usually people are put on secondary prophylaxis if they survive their reactivation. I'd like to turn now to some of the data on effectiveness of treatments that don't come from clinical trials. Isabella is going to cover the clinical trials, and I'm going to cover a couple of observational studies that have been really key in our thinking about treatment, especially of adults. So this is the first one. It came out in 2006 from Rodolfo Viati in Argentina. So this was a clinical trial, but it was not randomized and it was not blinded. So people knew whether or not they were treated, as did their physicians, and they were systematically assigned to treatment or no treatment. The second thing to point out is that this was very large. They had 566 patients, half of them treated, and the median follow-up time was almost 10 years. And the primary outcome in this study was progression of cardiac disease, progression from a lower severity class to a higher severity class. And they used what's called the Kushner classification, which is the first of those classifications. Got it. But I seem to do it about 5 times each time. This was, as I said, non-randomized and not blinded. It had about 20% loss to follow-up, and so it went through quite a rigorous review before it was published. They did sensitivity analyses to look at what the effect would have been if the people who were lost to follow-up had either progressed or not progressed, depending on which group they were in. But I think the bottom line is that they found that the treated group had a significantly lower rate of progression compared to the untreated group. And to me, this was, when it came out, very, very impressive. So 12 of 283, or 4% of those who were treated, had progression to a higher Kushner class. So that meant they either went from having no signs and symptoms to having an abnormal EKG, or went from having only an abnormal EKG to having signs of cardiac insufficiency, and death was also one of the outcomes. Versus 40 of 283, or 14% of those who were untreated. And this gave an adjusted hazard ratio, it was adjusted for ejection fraction, that was highly significant. So essentially, if you look at that hazard ratio, what it means is that the treated group had 75% less progression than the untreated group. The other thing that I think for those of us who think about the natural history of Chagas disease and who also think about clinical trials where an outcome might be clinical progression is that those who had disease already were much more likely to progress. So if you look at the untreated group, 7% of those who were indeterminate at baseline progressed to one of the higher classes. But of those who already had EKG changes, it was 19%. And of those who already had signs of cardiac insufficiency, it was 46%. And so that actually is part of the thinking behind the benefit trial, which we're all waiting to hear the results of, in which they recruited people who already had EKG changes as being a group that was at highest risk of progression. The other point that I want to bring up from this study is that the mortality rate was lower in those who were treated than in those who were untreated. And this did not reach statistical significance. The P was 0.085, but still, I think this is a pretty strong trend to decrease mortality. The other observational study that I'd like to bring to your attention was done by the group that Sergio is the senior member of in Argentina. And this is something that actually 8 years ago we put into the recommendations in the JAMA article that came out of the CDC group, that girls and women, non-pregnant women of childbearing age should be prioritized for treatment because we figured that it would decrease their chances of transmitting to their infants, but we didn't have any data at that time to prove that. And this study, to me, is a very impressive demonstration, even though it's not a clinical trial, it's a retrospective analysis. There were 132 mother-child pairs who were treated versus 222 mother-child pairs where the mother was not treated. None of the children of the treated women were infected, compared to 15% of the children of the untreated mothers. And to me, that was a really important finding, and supports what we had recommended back in 2007. The other question that's going to come up in all of the subsequent presentations, I suspect, is the question of what happens to serology after treatment. And so I just want to bring up some of the findings in these studies that I've just talked about. So in the Viotti study, negative seroconversion, conversion to a negative ELISA, occurred in only 15% of the treated versus 6% of the untreated. And I think that has to do with biological variability in these tests. And the median time to negative seroconversion was almost 12 years. In another study from Sergio's group in Argentina, so in the second line, up to 40% of those who were treated converted to negative serology versus none of the untreated, but it took up to 30 years. And then these data are from the same study of women, treated and untreated women, that I just showed you the flow diagram from. And this is only the treated women. And I found this quite interesting because we've always said this. Seroreversion, reversion to negative serology, was more rapid, or maybe we should say not quite as slow, in those who were treated as children compared to those who were treated as adults. So the dotted line are women who were treated when they were 15 years or older, younger, compared to those who were treated after the age of 15. And you can see that there was somewhat faster reversion to negative serology, but it still took years, more than 10 years for most people. And that's all I have to say today. I'm happy to answer questions now or later.
Yeah, why don't we take questions as part of the panel discussion, if that would be okay. So I think as we have our discussions this afternoon, it's very important, particularly for the panel, that we focus on our goal, which is to have drugs commercially available in the United States. I think our patients articulated what a challenge the current situation is for them. And to that end, I've actually asked Joe Turner to outline for us what those regulatory standards are and what our review consideration for a new drug would be. Joe is an infectious disease physician trained at Georgetown, was on the faculty at UCSD before joining the agency. And at the FDA, he's worked in basically anything in infectious disease I can think of. He started out working with antiviral drug development, moved to vaccine drug development, and we were very lucky about 5 years ago to recruit him to join us working on antibiotics, antifungals, and antiparasitic agents. Thanks, Joe.
Thanks, John. So in the next 10 minutes or so, I'll try to lay the groundwork for what FDA has when under consideration when we review new drugs. So my talk will cover adequate and well-controlled trials. We'll talk about endpoints and then examples of regulatory approvals. So this is a general overview of adequate and well-controlled trials. They're trials designed to show that a new drug is safe and effective for treatment. And by effective, we mean that it's the benefit that the patient experiences, a cure of disease, improvement in symptoms, and by safe, it's what are the risks of the side effects of the drug. And at FDA, and as well as clinicians, we always weigh the benefits and the risks of new drugs for treatment. So, and our statutory guidelines direct us that new drugs for approval must meet the standards for effectiveness and safety. And so this comes from Section 505D of our Food, Drug, and Cosmetic Act. And the Act was amended in the 1990s to include Section 115A. The Act talks in plural. It talks about trials, plural. The amendment to the Act, the Modernization Act, clarified that one adequate and well-controlled trial could meet the statutory standards for effectiveness and safety. And so the Food, Drug, and Cosmetic Act also says that the evidence must come from adequate and well-controlled trials, and our Code of Federal Regulations describe those. And so there are 5 types of trials that are specifically discussed in the regulations. And so the first example is the placebo-controlled trial, and it's where a test drug is compared with a placebo in an active preparation designed to look like the test drug. And success is that the test drug is better than placebo. And by success, what we're talking about is the statistical inference testing that shows a robust evidence of efficacy for the drug. The second example is a dose comparison trial, and it's where 2 or more doses of the test drug are compared. And success is that 1 dose of the test drug is better than the different dose. Usually it's the higher dose is better than the lower dose. The third example is a no treatment concurrent control trial, and it's where a test drug is compared with no treatment. Usually patients are randomized, the flip of a coin at the beginning of a trial to receive the test drug or to undergo no treatment. And success then is defined as the test drug is better than no treatment. The fourth type of trial design that's described in the regulation is the active treatment control. And it's where a test drug is compared to a known effective therapy called the active control. And success can be described as the test drug is better than the active control, or what we're used to dealing with in many trials of antibacterial drugs is that the test drug is similar, or what we call non-inferior to the known effective therapy. And in the non-inferiority case, we really have to have clear and convincing evidence of the treatment effect that the known effective therapy has over placebo. And the final type of trial design that's described in the regulation is historical control trial. And this is where a test drug is compared to experience that's derived from the historical literature or natural history of the course of disease. And then, of course, success is that the test drug is better than the historical experience. This is pretty rarely used in the regulatory setting, but the examples that are given in the regulations are, and the historical experience shows a very high rate of mortality, and the test drug then would result in a lower mortality. So with adequate and well-controlled trial is going to use an endpoint to demonstrate efficacy. And so now I'm going to go through endpoints and how we define endpoints. And the Code of Federal Regulations defines an efficacy endpoint that the method of assessment of subjects' responses are well-defined and reliable. The protocol for the study and the report of the results should explain the variables measured, the methods of observation, and the criteria used to assess response. Another definition of an efficacy endpoint comes from a Federal Register document from FDA where we say a clinically meaningful endpoint is a direct measure of how a patient feels, functions, or survives. And I'll come back to this Federal Register in a moment. The Biomarkers Definitions Working Group convened a meeting in early 2001 and provided this definition of an efficacy endpoint. And it's a characteristic or variable that reflects how a patient feels, functions, or survives. Clinical endpoints are distinct measurements or analyses of disease characteristics observed in a study or clinical trial that reflect the effect of a therapeutic intervention. Clinical endpoints are the most credible characteristics used in the assessment of benefits and risks of a therapeutic intervention in clinical trials. The Institute of Medicine also used this definition in their report of biomarkers and surrogate endpoints. And so what are types of endpoints that we think about in clinical trials? And I'll talk about 3. And the first is the clinician-reported outcome. And we're very familiar with this. It's an assessment of the patient's condition based on a clinician's observation and interpretation. And it has a lot of advantages. Often they're standardized, they're reproducible and consistent, and they're well-defined and reliable. An example in the antibacterial world is the reduction in the size of a skin lesion in a patient who has a skin infection within a couple of days, the reduction is observed by at least a 20% reduction. Patient-reported endpoint measures are any report of the status of the patient's health and condition that comes directly from the patient, without any interpretation by clinicians about how the patient functions or feels in relation to a health condition or treatment. An example that we've used in the assessment of efficacy is a patient-reported outcome symptom measure used in an inhaled antibacterial drug trials in patients who have cystic fibrosis. And now I'll talk about biomarker endpoint measures. And that same definitions working group defined a biomarker as a characteristic that is objectively measured and evaluated as an indicator of normal biological processes, pathogenic processes, or pharmacologic responses to an intervention. So usually we consider this as a surrogate endpoint, and it's rarely used as a primary efficacy measurement, but I'll give you an example. Another definition of a biomarker endpoint measure comes from our accelerated approval preamble to that new regulation. And it's a surrogate endpoint or a marker is a laboratory measurement or physical sign that's used in therapeutic trials as a substitute for a clinically meaningful endpoint that is a direct measure of how a patient feels, functions, or survives, and that is expected to predict the effective therapy. So the accelerated approval endpoint in 1992, that regulation was meant to address chronic disease conditions for which therapeutic intervention showed an important difference on a biomarker that was reasonably likely to predict clinical benefit and then could support approval and make a new promising drug available while additional clinical trials are ongoing to confirm the clinical benefit. And so some examples of biomarker endpoint measures are HIV viral load. And this is an example of a biomarker that has been through quite a bit of rigor to show that it's a direct measure of how a patient feels, functions, or survives. The second example is a drug for treatment of tuberculosis. The TB culture conversion to no growth is a biomarker that has a regulatory meaning, and I'll cover this in just a moment. And a third type of biomarker is serologic testing for antibodies to the parasite that causes Chagas disease. So now I'm going to give some examples of regulatory approvals, and there are really just two that I'll cover. And that's a standard approval, and that's an adequate and well-controlled trials have shown that a drug is safe and effective on the basis of a clinically meaningful endpoint. An example is a new drug for treatment of skin infection on that endpoint that I had just mentioned, the reduction in the size of the lesion. That's a clinician report. Second example is a new drug for treatment of HIV/AIDS that's approved on the basis of reduction in HIV viral load. And although it's a biomarker, multiple clinical trials enrolling tens of thousands of patients have validated this endpoint as being a primary efficacy endpoint that's used to approve new drugs for treatment of HIV/AIDS. Next is accelerated approval, and this is where adequate and well-controlled trials has shown that a drug is safe and effective on the basis of a surrogate marker, and the surrogate marker is one that's reasonably likely to predict clinical benefit. And for a drug approved and marketed under the accelerated approval, the sponsor is obligated to conduct additional trials to confirm the clinical benefit. An example of this is drugs for treatment of tuberculosis. They're approved on the basis of the surrogate endpoint of converting the TB culture to no growth, but then trials are ongoing to show that there's a continued clinical benefit and demonstration of cure of tuberculosis in patients. And so in summary, I've gone through some definitions of adequate and well-controlled trials that are show substantial evidence of efficacy and safety, and our regulations describe several types of trial designs. Endpoints are a measure of how a patient feels, functions, or survives. they're either patient-reported or clinician-reported outcomes. A biomarker is usually considered a surrogate marker reasonably likely to predict clinical benefit, although there are rare examples where a biomarker is used as an efficacy endpoint. And then we have a standard approval as a regulatory approval pathway. And we also have the accelerated approval where a drug can be approved on the basis of a surrogate endpoint. And thanks very much, and be happy to answer questions during the discussion session.
Thanks very much, Joe, and be happy to take questions once we get to the discussion. We've given Dr. Ribeiro the hardest talk to give. She is head of the Chagas clinical program at the Drugs for Neglected Disease Initiative, and she focuses on drug development for Chagas disease. She's worked in collaboration with industry and academia in the design, coordination, and overview of safety and efficacy of clinical trials of new drug candidates for the treatment of Chagas disease, and she's also conducted research and co-authored publications evaluating Chagas disease progression as well as blood-derived biomarkers. So thanks very much. She's going to give an overview of clinical trials which have been completed or ongoing or planned.
Thank you. It's a privilege to be here and presenting. A number of colleagues around the table have been involved with some of these trials and are involved in some of these trials. And just really, as indicated, providing you with an overview of where we are in terms of clinical trials for Chagas. Oh gosh, it's to the...
Yeah, he's going to show.
Oh, the computer will be here. This one. Very good. Thank you. So just not to belabor the point, but but it's important to frame the discussion in terms of Chagas disease as an unmet medical need. In terms of most common parasitic disease in the Americas, a leading cause of infectious myocarditis worldwide. We have 2 drugs available, nifurtimox and benzimidazole. But both of these drugs actually developed and registered in the 60s and 1960s, 1970s. Therefore, really with data that and those registrations that were based on small case patient series, small trials, actually with data that was generated from also at a time where you had a number of acute cases also. So it's important to consider the timing and the period in which registration was granted for both these compounds. And the situation today, as one discussed earlier, is that you have less than 1% of those that are infected receive treatment. And but it's not only because they basically, the large majority of patients don't get diagnosed, as the largest burden of the disease are patients that are asymptomatic. So, but in addition to that, there are the safety and tolerability issues, the long treatment period involved. So, but the resultant situation is one of really a great part of those that are affected are not receiving treatment. Oh, it doesn't show very well, I'm sorry for in the screen, but that I thought it was important to start by the systematic review from the CDC group, first authored by Karen. Barbara was also one of the co-authors, I think Luis Kirchhoff also. But actually at that time, this JAMA review actually showing that we had just 3 randomized controlled clinical trials and that had been published. And in addition, there was the observational study from Viotti actually assessing benzimidazole for treatment. But this is actually from a number of case series, a number of studies, but there were 3 randomized controlled trials, and this in chronic Chagas disease in published literature. Then in 2009, another review just basically showing the same. What you have is the same 3 randomized controlled clinical trials and a number of observational studies where one certainly reports the benefit and favoring, the benefit of benznidazole in terms of the different measurements that were assessments that were used, but essentially randomized trials we were talking about 3. And 2 of these trials, and again chronic Chagas. The first of these trials was it's one from Andrade and colleagues in a clinical trial in Brazil done in children, and where here serological outcome, serology was used as the endpoint, as the primary endpoint for evaluation of treatment response. And indeed, there was a significant benefit from benznidazole versus placebo. This was a double-blinded, placebo-controlled study, and where the ELISA was used, but also there was serological response over time, where also another measurement was AT ELISA, which is the so-called F23 or lytic antibodies. There are different names for this other type of non-conventional ELISA was used as an endpoint. But so indeed an important response, but no clinical outcomes in a population that is largely of children with early chronic infection, largely asymptomatic, but an important response and differences which actually, together with this other trial, that was a trial, an evaluation, also a second double-blinded randomized controlled trial in children, first authored here, Sergio Sosistani, in the group in Argentina, where again, a significant difference in terms of serological response in kids were seen. The results of these trials, these 2 trials, actually prompted the change in policy and recommendation for treatment of children by the WHO and by the number of different control programs. And in the study from Sosistani, from Sergio, there was also a xenodiagnosis. There was a parasitological measurement too, which was xenodiagnosis. And the small graph that you can barely see out there is actually showing that about in the order of 5% persistent negative, persistent positive among patients with benznidazole. But a significant difference across the board with both trials and benznidazole treated children. The situation in 2008 was one that you had 2 randomized clinical trials that were ongoing in adults with chronic Chagas disease. One, the Triana trial, the trial that involved chronic adult patients with chronic Chagas disease, with both in the indeterminate phase, but also with target cardiac involvement, and the benefit trial. I'll talk a little bit about both trials later on. So the benefit trial here, a double-blinded, placebo-controlled trial evaluating benznidazole treatment in patients with early cardiac involvement. But by then, in 2008, the situation was that really had decades of no new clinical trials for treatment options in Chagas. And research and development really stalled by really a number of knowledge gaps. And one of the essential and what key gap here was actually how to evaluate treatment response, particularly in adults, where you have this very long treatment, very long disease evolution period, where the large majority of patients are asymptomatic, and so really how to assess treatment response in such scenario. This besides a number of other knowledge gaps that were in technical challenges with the disease. So there were a number of discussions, expert groups that were in the Chagas disease platform, clinical research platform. There were groups that got together to really see that, well, as there is a clear need for new treatment options for patients with chronic Chagas disease, both adults and older children, how should we go about? And so a decision was really to proceed with clinical development and generating scientific information that will help fill existing gaps and inform also future drug development. PCR was selected as the primary endpoint for these clinical trials after a number of consultations. And there was at that point a standardized methodology that had undergone multi-center evaluation. Dr. Schiffman will discuss this a bit further. And the consensus was that as there was concern with the low sensitivity of PCR, that one would aim for serial and multiple PCR examinations done sequentially. And the rationale for selecting PCR as the endpoint was one that was a plausible biological rationale with the link of parasite persistence with chronic heart inflammation, data from animal models in that support. There was also human data from acute Chagas disease in children, and also from reactivation, Chagas disease transplants, and HIV. And also some information from observational studies. There were early regulatory consultation in the region and then agreements in terms of endpoints, trial design, and strategy for development. And that we were aiming to, in this process, also to generate pharmacokinetic, pharmacodynamic data in humans using the different available biomarkers, doing parasite genotyping for new candidates, but also for benzimidazole in particular. So there has been progress over these recent years. These discussions actually took us forward. Two studies of population PK studies in children were carried out. Until very recently, although kids, as we discussed before, there was a recommendation for treatment of benzimidazole in kids, and there was no pediatric formulation, but there was, beyond that, there was no PK information in kids whatsoever. And so actually, there was data generated in children on pharmacokinetics in kids. The azole class of compounds that for 15 to 20 years had been considered as very of a class of compounds with a lot of potential for Chagas. Actually, three clinical trials were initiated. The Chagas-azole trial, Stop Chagas, that is a Merck-sponsored trial on posaconazole, and also E1224 and benzimidazole in adults, a phase 2 trial in Bolivia, and more recently, fexinidazole, another nitramidazole for Chagas. So quite a number of new information being generated. So I'll talk a little bit about this trial. So in the case of, this is the Chagas-azole trial, results of which came out in 2012. You had the publication in New England Journal last year, and it was one cannot emphasize how surprised most of us were when we actually saw these results. Because posaconazole, basically 2 doses of posaconazole were tested. We had also evaluated, this was the team from Israel Molina in Spain. These were patients with Chagas, adults, Chagas in the indeterminate phase. Most of the patients, they were actually Chagas with chronic Chagas disease. Some patients actually had heart disease. And basically, this was randomized. The trial was randomized, and you had 80 to 90% of patients randomized to posaconazole failing treatment at 12 months, and actually 6% of benzimidazole treated actually failed at 12 months. So basically, the primary endpoint for this trial was PCR. It was actually sustained parasitological response at 12 months. So all patients had cleared parasitemia measured by PCR, had cleared circulating cruzi DNA at end of treatment, and at 12 months, as indicated, 6% of benzimidazole treated patients had failed, so 95%. had actually had sustained response. So this was actually a surprise, the failure rates that were seen with prosaconazole. Another study, this one is a prodrug of ravuconazole, it's 1224, and this is a trial comparing 3 different doses and durations of treatment with a 1224, a benzimidazole arm, and this was designed as a double-blinded, randomized, placebo-controlled, randomized clinical trial. So a lot of information on this slide, but essentially a lot of new information here. At the end of treatment, at day 65, when you look at benzimidazole basically, at the end of treatment, you had 90% of patients had cleared parasitemia. The same across, no difference across the different treatment arms on E1224, but basically at 12 months follow-up, you had 81% of benzimidazole treated patients had sustained that response, and with about 29% in the case of E1224, a high dose. The low dose of E1224 had no difference from placebo. But here again, the primary endpoint that was selected here was actually PCR. We actually had, in contrast with the trial from Molina, we actually did 3 PCR examinations at each one of the time points. PCR was evaluated during treatment and at end of treatment, month 4, 6, and 12 months. But what was quite interesting also from this trial is that if you look at the graph on the side, where you actually have the results of the PCR during treatment, is that within 7 days of initiation of treatment with benzimidazole, you actually had already a very significant difference in terms to placebo. Placebo is the flat blue line on the top, and actually at 7 days you already had benzimidazole here. With 2 weeks of treatment with benzimidazole, you actually had you were below the limit of quantification for PCR, and there it stayed throughout the treatment period. And then if you follow until 12 months, you actually had, so even among benzimidazole failures, those actually you had this sustained, very significant difference from placebo over the 1 year of follow-up. And so this is actually the first trial that actually did the PCR treatment response during treatment. And we actually had some interesting results actually in adults. And so this was quite interesting to see how early the treatment response was and how this was sustained, and actually even in those that failed, how different the PCR results were from placebo. In the case of azole, of E1224, you actually had, with the high dose, you actually had this drop. Also, across the treatment arms, with azoles during treatment, you had this important response. But as soon as treatment was discontinued, you actually had the slow return up. The 2 lower doses of E1224, there were no difference from placebo. But that actually, in the case of the high dose, it remained different from placebo. Lastly, and I'm sorry, here you also don't see too well, but we actually did the AT ELISA, and so therefore it's the lytic antibodies that were also done in kids. And it was it's interesting to see is that actually at 12 months there was a significant difference from placebo, also demonstrated in adult patients with chronic Chagas disease. So with regards to conventional ELISA, we saw no difference. So I'm sorry for this now at this point, just to talk a little bit about the public case studies in kids, the 2 trials, one in kids from 2 to 12 years old and one from newborns to 12 years of age. And kids, actually, most of them, this is actually single-arm prospective puppy case studies. Dr. Alce as the first, as the principal investigator. And what was very interesting on these studies is that actually, in both, you had 100% PCR negative at end of treatment. But what one looks across the blood levels, across the blood concentrations, benznidazole concentrations in kids, is that the lower the age group, the lower the blood levels, and there is a really, when you have patients that are 7 to 12 years of age, there was really no significant difference in drug concentrations from adults. But actually, that kids below 7 years of age actually have a significant difference from adults. So, but they still have actually 100% PCR response at end of treatment. And so it raised the question is, have we been overdosing adults all along? And would that be one of the explanations actually for the safety and tolerability issues that have been at least partially responsible for that? Importantly, in some of the pediatric cohorts also from Children's Hospital in Buenos Aires, from Dr. Alce, which actually is this indication that not only the PCR response at end of treatment is actually 100%, but actually that's actually sustained for patients for which data is available beyond 12 months, is that actually, it stays down. It's not that patients are actually becoming positive again, that you actually have late relapses. In this other graph here, it's actually looking at ELISA, conventional ELISA over time, and also F2/3 of lytic antibodies over time. So it's really giving an idea of different outcome measures and how in some way this PCR it's really showing an early response outcome assessment. I will not discuss this because of an issue of time and also because it doesn't show too well. But it was actually just in terms of providing additional information in terms of the clinical trials and this very different clinical trial scenario that we have today. So Stop Chagas, not a clinical trial. This one is a Merck-sponsored trial. Dr. Murillo is not here today, but he's one of the PIs. And evaluating actually different doses of posaconazole versus placebo, and actually has a benznidazole and posaconazole combination arm. And again, across both the Molina trial, E1224 trial, and this one, as you can see, the design was very similar, where the primary endpoint is actually, for all these trials, were actually PCR selected as the outcome measure. We evaluated response at end of treatment, but also followed patients for to complete 12 months in looking at sustained parasitological response at this point. This trial has actually recruited 120 patients. Enrollment and follow-up has been concluded, and we are expecting actually over the next month for the results to be available. The Traina trial I mentioned very briefly before. I have a single slide because there is analysis that is still ongoing on this trial. It's a very important trial. Patients with adult patients with chronic Chagas disease, indeterminate phase, and also with cardiac involvement. This is randomized, placebo-controlled clinical trial with actually follow-up of patients over 10 years. So this trial actually, one has serological response, PCR, and also clinical outcomes, with a follow-up of basically in the order of about 10 years. As you can see, 910 patients underwent screening, 764 patients were randomized, 382 placebo, 382 benznidazole. About 300 completed study in benznidazole arm, 308 in the placebo. And what you see, and here the graph, basically you have this very significant difference in actually the placebo, in the benzimidazole versus placebo response in terms of PCR outcomes and also serological response over time. And here we looked actually at sustained parasitological response measured by PCR at 12 months. And actually when you look at the benzimidazole response, it's 86% sustained response. So it's really looking at this pattern of quite similar in terms of when one looks at the benzimidazole data across the different studies through the different outcome measures used. Benefit trial is a key trial that we all awaiting the results. There is a pilot study that was evaluated that has actually a co-primary endpoint of actually negativization of PCR and also an evaluation combined actually with the reduction in the mean burden of the parasite load over time by PCR per treated group. This is a randomized double-blind clinical trial, adults with chronic Chagas disease but with early cardiac involvement. And it's really the follow-up, the mean time of follow-up follow-up of these patients in the order of about 7.5 years, and the trial is finishing now. So we are expecting now the last visits are being made this April, and they're reporting in the order of 1.5 lost to follow-up. And for the main trial, what you have actually is the primary endpoint is a clinical benefit endpoint. So here you have 2,856 patients randomized equally for benzimidazole and placebo. And the primary endpoint, it's really evaluating a combination of clinical outcomes of death, cardiac arrest, resuscitation, sustained VTAC, need for pacemaker or defibrillator implantation, thromboembolic phenomenon or hospitalization for heart failure or heart transplant. So this you have, it's also a trial that is multi-center, multi-country. You have actually the countries Argentina, Bolivia, Brazil, Colombia, and El Salvador, actually with another. So it's actually also taking into consideration the impact of the genetic diversity. So as you can see, the large majority of patients in the benefit trial, actually, it's 75% is really New York Heart Association Class 1. So it's really early stage, and you have actually about still about 14 patients that actually from medical history had already gotten pacemaker, but still the large majority early disease. The interesting data in terms of early on from drug compliance, you can see that very few patients, you had 16% in Argentina, 12% in Brazil that actually discontinued, had treatment interrupted at some point. But you have very large and very good compliance overall in terms of treatment. So it's going to be this really, we're very much looking forward to the result. And this is not unblinded yet. So you can see the PCR baseline, it's in the order of 60%. Only a single sample, PCR sample was done. So this is actually one of the things that is interesting, is that here you have 60% with a single sample. In the case of the E1224, for example, we had in the order of about 86% PCR positive at baseline, all right? And so this is actually looking at both placebo, this is actually the pooled integrated results of PCR over time. And so not yet unblinded, but shortly to be done. This, just a few notes, and just to conclude, a couple of clinical trials that are also ongoing. You have fexinidazole trial. It's evaluating this new nitrimidazole, different treatment, different doses with different treatment durations 2, 4, and 8 weeks with the low dose, and the same with the higher dose, and a matching placebo. Again, the same pattern, evaluating response during treatment, end of treatment, in this case up to not 12 months, but actually 6 months for proof of concept. This trial was recruitment was interrupted. We have 47 patients randomized, and we are actually in the midst of evaluating the response. We have, and there is this planned trial to be initiated now in October of this year, October, November of this year, where we are actually testing the hypothesis that we discussed before. Can we actually shorten the treatment duration with benzimidazole? Can we lower the dose with benzimidazole? Would combination make a change? And so this trial actually looked at new doses and duration of treatment with benzimidazole, and also combination of benzimidazole and E1224 in adults, adult patients with chronic indeterminate Chagas disease. So in terms of future clinical trials in chronic Chagas, there is a planned study of benzimidazole in kids and children. There is a trial sponsored by ELEA Chemo Group with the Mondosano Foundation. It's actually assessing efficacy and safety of benzimidazole in children. And the design is under finalization, it's under discussion. There are 2 trials on new benzimidazole treatment regimens in adults that are planned. One, the DNDi-sponsored trial in collaboration with ESAI, ELEA, Mondosano, and also the Berenice project. Again, in this case, PCR as the key treatment response, and also a response on an evaluation with lytic antibodies. There is the Berenice project, as I mentioned, and then there is the nifutimox in children. This one, a Bayer-sponsored trial of efficacy of and safety of nifutimox in kids, and includes both PCR and serological, but the primary efficacy response in terms of serological response over time. Reduction of titers. I would not discuss these results, but I thought it was important. Dr. Byrne, Karin Byrne already discussed these, but it's really very important data from Sergio Sosistani and his group in Argentina in terms of the impact of treatment in actually of women in preventing the transmission of congenital Chagas. And this data is now corroborated. There is work from Dr. Alche that is being published now. Again, in this case, 394 women, and actually 15 pregnancies, 16 children, and again, it really corroborates the finding from Dr. Sosistani, in which really has there were no transmission, there was no congenital Chagas documented post-treatment of girls and women of childbearing age. So just, I will not take you through the whole design of this. This is actually a non-human primate study that is ongoing, looking at how PCR response in the blood correlates with sterile cure. This is a study actually that is done in naturally infected monkeys, non-human primates, and that are treated and followed over time, treated with benzimidazole at different doses, treated with placebo, and treated with E1224. And basically, following treatment, animals are actually, after 12 months of follow-up, they are immunosuppressed, and after immunosuppression, animals that do not show reactivation will be actually sacrificed, and then one can evaluate tissue, the presence of parasite in the tissue. So it's actually to provide data in support of PCR and the rationale for use of PCR as a marker. So the Chagas landscape has changed significantly with some of these discussions and these decisions that were taken forward. But there is a lot to be done. I think that fundamentally here, and I come to conclude that there was a significant impact of this recent clinical trial data, both in adults and children, in the overall Chagas disease R&D landscape. This data, both on the azoles, but also on benznidazole, key data on benznidazole, actually leads to a push for scaling up diagnosis and treatment in Chagas, also for improved access to available drugs and formulation, but also points to the use of these markers, including PCR, in terms of for evaluation of treatment outcomes, treatment response in Chagas. There is really work to be done towards new treatments for the chronic form of Chagas, and we need to continually generate and analyze data on pharmacokinetic and pharmacodynamics for new treatments in Chagas. And we're looking forward to the results of the new proof of concept studies on new treatment regimens for benznidazole in monotherapy and in combination. But it's clear that we come to a clear regulatory framework for registration and marketing authorization of new but also old treatments for patients with chronic Chagas disease. And here we make the case as we come forward and linking to some of the discussion that we had just before that if PCR or serological response, if it will be actually could be used as a basis for registration of compounds for Chagas. The situation in kids, in children, it's simpler somewhat because of the timelines for follow-up. In the case of adults, where you have this very long period for demonstration of clinical benefit, this is really particularly challenging. But this is it. Thank you very much.
Great, thanks to thanks to all of our speakers for excellent talks. We're going to begin the first panel discussion. Sumati Nambiar is going to be moderating those panel discussions. Sumati is a pediatrician who practiced pediatrics for some time prior to an infectious disease fellowship at the Children's National Medical Center here in Washington. She joined the FDA, and we were delighted when she was appointed director of the Division of Anti-Infective Products a short time ago. That division regulates antibacterial, antifungal, and antiparasitic drugs.
Thank you, John, and thank you, Dr. Ribeiro, Dr. Byrne, and Dr. Turner for your presentations. Very useful to get the discussion started. So I think as John had mentioned when we started this afternoon's discussion, really the task at hand is how can we get safe and effective therapies available for our patients. And I think as Dr. Ribeiro mentioned, how can we have a reasonable regulatory framework such that these products can be developed? And we've heard this morning from patients the difficulties they're encountering because these therapies are not readily available. So to that end, we've sort of split the discussion into two parts. And the first panel discussion before break, and I know we're running a little late, so we'll try and maybe wrap this up by 3, around 3? 20 minutes. 20 minutes. Okay. So we'll try and focus on what populations we could do these clinical trials in and what might be some acceptable control groups. We'll take a break and then we'll come back and talk about trial designs, endpoints, proceeding which we will have a presentation on laboratory monitoring using various methods. So, you know, we've certainly had a lot of discussion internally on these topics. We have been impressed by the interest in developers, you know, trying to certainly bring new products, but also trying to make the existing products available in the U.S. market. So one important point and one important topic I think we would love to get input from all of you is what might be the appropriate patient populations. You know, certainly we could study it in acutely infected patients. We could study it in children, maybe children with congenital infection, where the numbers are probably going to be smaller, but there are some benefits in that you might be able to assess the outcomes at a shorter time period. Or do we focus on patients who are in the indeterminate category where we have a larger number of patients, But the problem there is the follow-up tends to be much longer unless we are now willing to adopt some of the newer methods such as PCR and what are the uncertainties around it. So I would welcome input from the panel members. I think one key question is, and I think this came up in Dr. Byrne's presentation, the number of patients with Chagas disease who are really diagnosed in the acute stage of the disease is very small. Is it feasible, is it practical even to do a study of any reasonable size in that patient population? So that's one, certainly one question we would like to ask. The second is, what are the pros and cons of trying to study a compound in the indeterminate sort of group, which is probably the largest number. So, yes, Dr. Mehman.
May I ask a question as a non-scientist clinician?
Certainly.
We are currently using two drugs, nifurtimox and benznidazole. Neither are FDA approved. We're rather meticulous with our use and our follow-ups, and there's ample data that they're effective. They're reasonably effective. What do we need to do? We're using these drugs currently, but it's restricted. What are you looking for in terms of a study design trial to show what that we don't already have? Because I'm really confused.
Because I think a very valid question. I mean, I think as Joe had presented in his slides, we need evidentiary base. We need a basis to be able to approve these drugs to say they're safe and effective. So what is the evidence? And I know there is a lot of clinical experience. We certainly have studies done by doctors, that we've discussed. We have first authors of these studies in our midst. So certainly one consideration that has been given is if we in fact have access to the primary data, the data that actually supported these studies, is that something we could consider? But I think our biggest issue is the quality of the data. So we just cannot approve drugs based on a publication.
That's what I'm trying to understand. Right. What is it that we're trying to do? Because you don't want to recreate the wheel?
No, we don't. So if we are able to get primary patient-level data...
And you're appreciating my level of frustration as a clinician.
Yeah, certainly. No, I think we are aware of it. And if we can actually get patient-level data, say from some of the studies that have been done, which clearly demonstrate the treatment benefit, I mean, that's certainly something we can consider. I think there are instances where we've done that, right? Correct me if I'm wrong, but
And do we have access to that data? Where we don't have to recreate the wheel and do another study design and looking at things that have already been done? Yeah. Pragmatically speaking.
Right, yeah. I mean, your question's a good one. And we were actually talking about this as the slides were going over. There's a lot of data out there, okay? To actually get a drug approved, somebody has to submit to us an application that has the clinical safety and efficacy data and also provides information on the manufacturing of the product. So, we don't make the drug, somebody else does. So all these pieces need to come together, but the question you're asking is the same question I think that we're asking ourselves too, which is, who has this data? Can we identify it? Can we get at are there some primary source records out there that might be able to be audited so that we can understand the quality of it? And then that should be paired up with who manufactures the drug. And then is there somebody out there that can submit an application that can manufacture this product, day in, day out to a quality standard so that it can be approved and it can be available? So does that help to sort of...
Yeah. And in that direction, we have companies who I know are interested in bringing these drugs forward.
Yeah.
And we have representation here from those companies. Is this something that you guys can get together and move forward? Or do we have to start at the beginning and redo everything? Because that's insane.
Dr. Byrne, do you want to make a comment?
Yes, I think that's a key question, obviously. I think that, and I think that if we discuss with a number of the investigators that were involved in these clinical trials, I have no doubt if that actually you're going to have the actually making this information available for registration of benznidazole, for example, or if But in the case of benznidazole, where actually you have the placebo-controlled comparisons done, etc., I have no doubt. I think here we have indeed representatives from ELEA, from Mondosano Foundation. There are groups that are collaborating this, and certainly this information can be made available. The issue, though, it goes back perhaps to a discussion that we're going to still have, is on the issue of the clinical benefit demonstration, right? If one would be able to actually register or seek marketing authorization here based on the outcomes that were selected for this. A number of them have serological response, but some have, but now we have also PCR data, and it's actually if one would be able to support such a filing based on the outcome measures that were selected for these trials, both either as a surrogate marker or as a primary efficacy endpoint, and I think that's it.
Right. So that again will depend on how good the data are. You know, if one really has to demonstrate a link and make a link between the benefit on the surrogate endpoint translating to a clinically meaningful benefit.
I'm a positive person,
yes,
and I'm looking that the data is good.
Sure.
And let's start with there instead of again recreating the wheel, because these are two drugs that we're using that we're seeing effective treatment. So just again, from a clinician's perspective, let's make this easy. We want it to be safe, obviously. I think everyone here who treats patients has patient safety at heart. And if we can do it in an easier fashion than recreating another study design and starting with another patient population that hasn't been treated yet and having a placebo arm versus a treatment arm and spending 5 years, 10 years doing that, you know, how many patients we're going to lose in that process.
You know, a very reasonable goal, and I think consistent with our thinking on this too. And the real question is trying to gather up the data and identify those that have it and see if they can essentially, join up with a manufacturer so that, there is a way to try and take advantage of all that's been done and agree why recreate the wheel if the quality data are already out there. So it's a very reasonable idea, and I think one that several of us are having, and the question is pulling it together.
Excellent. Thank you. Done.
So I guess we can take a break.
Well, we're not done. So I think that kind of answers the question on benznidazole and nifurtimox in terms of what the regulatory pathways might be. And part of our goal of inviting some of the key investigators here was to sort of show them that we really will need access to their data to have the drug approved in particularly in the United States. So that's great. I think we had written some of these questions thinking about, for example, a new azole. So what would be helpful to us is sort of focusing on a discussion, pretend this is a new azole or a new class. How would you design the trial? And we've certainly got the right folks here to answer that question. And what population would you do it in?
Yeah,
Well, about populations, we need pediatric formulations, then we need studies in children. Children, if you see different presentation, chronic disease begins after 2 months of infections. Then children present chronic phase of the infection. Then children have to be included in all of the studies that we began about new drugs. This is very, very important. This is one. We have a lot of non-treated children that now are adults. This is the central point. We have to think about that. And about control groups, this is very difficult to non-treat a patient when you diagnose these patients, because we have a lot of information about efficacy of benznidazole and nifurtimox. Then we we cannot use placebo for the studies.
If I can ask you a follow-up question. So if you are trying to study children, are these children who acquired the infection transplacentally, so these are congenitally infected children that you're picking up month 1 or 2, or are these children that get infected later in life, or do you differentiate between the two?
It depends about places. In places without control of vector infection, you have children infected, older children with acute infections. In big cities without vector transmission, you have a lot of congenital infected patients.
Thank you. Yes, Dr. Sosistan.
Yes. First of all, I would like to express my completely agree with the clinical point of view of Sheba. And regarding specifically a question, I think that several presentations put on the table the complexity of the natural history of Chagas disease, that is an infectious disease, a chronic infectious disease, and we have a permanent expression of infection and a very, very late clinical expression. If you think in these two concepts, I think that, as Jaime said, there are enough evidence of trypanosomide effect, benefit. And there are some evidence of clinical effect. A rational way to assess response of trypanoside treatment is on the infection, the rational. Because personally I think that after benefit trial, us to demonstrate clinical effect is unreasonable. Because it's not possible as designed to demonstrate clinical effect after benefit trial. So in this point, I would like to reinforce that it's necessary to design clinical trial to demonstrate effect against infection. Because in addition, we have demonstrated that the effect against infection can interrupt one of the main routes of infection as congenital transmission. And regarding the control, I think that in fact there are enough evidence that maybe in general would be not recommended use placebo arm, but in some cases, thinking that it's in chronic infection, it's possible maybe in some special situation, thinking a control, Gilead control, yes.
Just to answer the specific questions from the panel, I think that we have dwelled quite a bit with regards to the populations in which a clinical trial would be feasible and acceptable. And I think that we have selected the chronic indeterminate patients as they represented the highest burden of the disease and where there would not be an issue in terms of recruitment for one side, but also we felt it was important to actually have a placebo control, and we felt that that population would allow us to be able to have a concurrent placebo control and a double-blinded design, and therefore and that was considered acceptable, actually, as it would represent, as Sergio was indicating, delayed treatment. And so I think that actually the evaluation of acute cases today, it's not feasible.
I think that you know, the incidence is such that, and it goes so fast that we'll not be able to identify cases. Oral Chagas disease, actually the number of the clusters of oral Chagas are becoming more frequent, and or at least there is some indication to that, but it's episodic. You cannot predict where they're going to happen. so this is a consideration. But the other, it's certainly kids. Should we be, so certainly the children as a population for evaluation. I think there is one element here, though, is just that in one side there is recruitment, as you have pointed out, but there, as Jaime Alche indicated, I think there are indeed a number of untreated children so far, unfortunately, and therefore these trials are certainly feasible. But there is the question, though, is if the findings in kids will be representative of an adult population. So if one carry out clinical trials in the children, would the outcomes reflect what one would see in the adult population? Would the adults being the worst case scenario in a way? So here is where it's actually balancing these two sides, which is really challenging. And so it's a long answer, sorry for that.
So you think these children are identified as part of screening when serologic screening is done routinely in some countries? Is that how they would be picked up?
Okay. Particularly, for example, countries like Argentina, Bolivia, where you have actually antenatal care mothers are actually assessed, and so routinely also it's evaluated in mothers that are Chagas positive.
Yes, doctor.
Yes. I would like to add regarding the design of the clinical trial. We are working for several years looking for an appropriate design. We are really very, very happy to understand and to know with evidence that we can design short clinical trial in comparison with the past. In the past, we needed at least 5 or 6 years to demonstrate efficacy against infection. We can demonstrate the same effect in 12 months.
Right. You mean because of the availability of PCR? Is that your?
Okay, Kiliana Suzafroshnik Bayer. Just a comment. So we did not discuss about the trial design, but my colleagues are trying to put one protocol in place, and we have a problem of loss to follow up. And especially in studies with children with a disease that does not have a phenotype, an acute phenotype, it's very difficult to, after a 60-day treatment, to have a long follow-up about 1 year, 6 months, full at 12 months, because these poor families will be moving around, so small countries, and the possibility of that we have to maintain a full follow-up is almost known. I mean, we have been thinking in creative ways, but it's impossible to predict. And this causes risk for us, so that we cannot guarantee from the beginning that we will achieve 100% 1 year follow-up in order to have our data valuable. So this is a short comment.
Thank you.
I think we saw the results that we saw here. Yeah, I think what I was checking, most of the results show at least 10% or more loss to follow-up at 12 months.
And may I kind of add on that comment? I think besides that, you have also reinfection, that kids or family went back to the endemic area that you have no control about, and then they came back with reinfection, and it's very hard to tell. So it's a very difficult trial to design, actually, to conduct.
Yes, Dr.
I think it's worth noting, I think the experience that we had in the clinical trials in Bolivia, where we actually had very good rates of follow-up, I should say, and we actually had at 12 months like 3% loss to follow-up. Really very little missing data. It's very difficult, no doubt. The issue of reinfection is it's challenging. we actually had linked with the control programs, and in order that patients were informed, they needed to inform if there was reinfection and so on. But it's certainly a question, and that needs to be there.
And regarding the control group, it's always hard because, I mean, you ethically have to treat every single patient, especially in those areas that the drug's already proved for the last 40 years in available drug. And how can you not treat those patients, especially trial conducted in the area, unless you have a very chronic case, and then you are, the recommendation is different.
It's just to reinforce the comment of Isabella regarding the control on or exposition for reinfection. That is, it really depends of the size of the trial, of course. In some cases, it's absolutely possible to design a clinical trial with patients without exposition, or because they are living in area without vectors, or because they are living in an area under surveillance with interruption of transmission. That is absolutely possible. In fact, the control programs are demonstrating this status. And if the trial is huge, like benefit trial, we discussed a lot about that. But we assume that the eventual exposition is balanced between patient with besnidazole and placebo.
I want to ask Sergio if the data from the original data from your randomized study would be available to be part of a submission. Like by source data, I mean like the records of each patient, the laboratory results of them, or some kind of records when that study was done, the randomized and controlled study in children.
Yes.
Thank you. I think that will be very important because sometimes in the article, when you report, mean GMT reduction or something, like, median or what was the actual value, that those questions will be very important. Thank you.
All right. So thanks very much for that discussion. Just one point for sponsors that may be interested in developing nifurtimox or benznidazole. The FDA, following approval, publishes, makes available its reviews on the Drugs at FDA website. So one application that may be worth looking at as a recent precedent for reliance upon efficacy trials that you yourself did not conduct would be miltefosine, approved recently for leishmaniasis. So that may be worth taking a look at. Chagas is challenging in that it's likely that we're going to need to understand better the surrogate endpoints that were used in the trial. By now you know what the FDA calls a surrogate endpoint, so that's good. We're using the same language. We're going to focus on that after the break. We're going to take a break now for 15 minutes and reconvene at 3:10. Thanks.